Spade, Dollar Babe, Marvin Way, Isabella, Limestone, Lorium, Barbo, Lucky Fella. Been everywhere, man. I've been.
Hi, this is Jim Islip with MSU Extension, and this is What's Up at UPREC on February 24th, 2021. Today we'll visit with UPREC Director Dr. James Dedecker about the Solvita soil test and measuring soil biological activity.
Hi there everybody, my name is James Dedecker here at UPREC, and today we're going to be talking about soil health and specifically the Solvita soil test that gives us an indication of our soil biological health. Soil health refers to sort of a holistic look at how our soil functions and how it can support crop production. Well, soil health not only considers that sort of chemical aspect of soil fertility, but also things like the physical and the biological aspects of soils that are important as well. So Solvita is actually a measure of soil biological health in that it measures CO2, carbon dioxide, that microbes in the soil are respiring or basically breathing out as they digest or decompose organic matter and also help to mineralize nutrients like nitrogen in that process. So we've got a few different soil samples that we're going to be looking at today, and we're going to go through the process of what it takes to run a Solvita test, and then also look at the results that we found on the example soils. These are examples of Rudyard silt loam from a farm out in Dedecker that we've been partnering with on an NRCS Conservation Innovation Grant. It's fairly fine in particle size with quite a bit of silt and also some sand and clay making the loam soil. We've got a difference here between these samples of Rudyard silt loam in management. The farm that these came from is primarily a beef cattle operation, and so they are doing activities like raising hay, growing pasture, grazing, and then feeding out stored forage in the form of hay that is fed on the pasture in a process called bale grazing, where the bales are actually placed out in the pasture and the stock are allowed to graze them, depositing waste hay and manure strategically around the farm to address issues like soil health or maybe erosion by putting more residue on the soil surface. The first sample that we have here is an example of a field that has been in hay production. And shortly before we sampled, that field was tilled to be renovated and grew back to Timothy. So this is a hayfield situation that hasn't seen grazing, and particularly not bale grazing. As we move over to the next sample, this was also a new seeding, but this was an area that was historically pasture. And then the pasture was recently renovated with a new seeding, so there was some tillage and then replanting that occurred prior to sampling. This was a new bale grazing area, so an area of pasture that had been grazed historically but not seen this bale grazing. And so it was in bale grazing for a shorter period of time. And then over here we have a more established area of bale grazing where that practice had been in place for quite some time. When you look across these soil samples, you might notice some visual differences. The first thing that's pretty evident to me is a difference in color, particularly on this far sample where we have more history of bale grazing. Quite a bit darker. So right away, that's an indication that there might be more organic material in that soil as a result of that practice, perhaps. The other thing is, as you look closely at these soils, you can actually see bits of organic matter. That could be waste hay, it could be plant residues, it could be bits of feed and forage left in from the manure that was deposited on those sites too. Now when we sample, of course, we try to clear off the surface residue and organic matter to get a more accurate sample, but there's a lot of residue in these soils from some of those practices. So certainly where the bale grazing is happening, we tend to have a little darker color and more residue. Solvita is a biological soil test. So there's a couple of things that can influence the outcome. One, we have to have the food in the soil for the microbes to feed on. And then we have to have the microbes present in a sufficient level to be active and consume that organic matter and release the carbon dioxide. We've got a few basic tools that are required for the Solvita test. Most important part for Solvita is actually the Solvita Dedecker probe itself. So this is a probe that is designed to be sensitive to CO2 concentrations, and it changes color based on the amount of CO2 in the atmosphere, in this case inside the jar. And this is an indication of what that color change is supposed to look like, where the probe starts out blue, and as the CO2 concentration increases in the jar and over time, this is a 24-hour test, you can see the color lightens to more of a yellow color. Solvita comes with very detailed instructions, and it's important to follow those to try to get as accurate of results as possible. There's a few different ways that Solvita can be used. One is for what's called basal respiration, and that would be if I took a sample of soil from the field at whatever moisture it happens to be that day, and I go ahead and measure that for 24 hours, and that gives you a sense of sort of a snapshot of what the conditions are in the field at that moment. And the CO2 burst method is different in that it's designed for use on dried soils, and then we re-wet them to sort of wake that bacteria up, and when that occurs, they release a burst of CO2 that can be measured. And also allow us to predict biologically mediated processes in the soil like nitrogen mineralization. So aside from the paddle, the tools otherwise are pretty basic. Solvita sells a jar that you can use, but you can also use just a regular 16-ounce glass canning jar. So the only thing you have to watch out for is making sure that the seal inside is in really good shape so that it seals properly and traps that CO2 inside the jar so it can be measured. We also have a small beaker that is used to contain the soil sample inside the jar. We've got some distilled water that we're going to add to the soil sample here using our pipette. This is a fancy one that allows us to accurately measure, in this case, 10 mils of water for each sample. The test requires 30 cc's of dry soil, and so Solvita provides this handy-dandy little cup for measuring that. The final piece of equipment, which is actually not required for Solvita but is really nice to have, is the Solvita reader. So when we run this test, we can just look at this colorimetric card and get a sense of the result. But there's only so many gradations that our eye can see, whereas this colorimetric reader will give us actually a number to put to that color, and then also give us the amount of CO2 that that corresponds with. So this is a much more sort of quantified way of getting more accurate results from Solvita. Okay, so the first thing that we need to do to actually run the Solvita test is to measure out the appropriate amount of soil. So here's our 30 cc scoop. We did a little bit of pre-processing on these soils. We first ran them through a 5mm sieve, and then we simply allowed them to air dry. And you want to be less than 3% moisture if possible. So we've got our level scoop of soil there. We're going to put it in this small beaker. Again, the wetting is pretty important. We don't want to over-wet some portions and under-wet other portions. We want to kind of add the water slowly to allow it to disperse through. Get our first 5 mils. The term that they use in the instructions is to dribble. So basically just spreading the water around here. Spread evenly wet. Then we'll grab our Solvita Dedecker. It's in this foil pouch. Important to realize that they do expire. They also should be stored in the fridge for the longest shelf life. This is what the paddle looks like. It's a gel type material that detects the CO2. You can see there's a little bit of liquid on there, but that's okay. You don't want to touch the gel itself, so hold it on the plastic portion. And we're going to stick it in the so. Then this all goes in the jar. Solvita does provide plastic mini beakers, internal beakers. We are using glass here today, and glass on glass is a little bit tricky, so be careful that you're not swinging it around or cracking the two pieces together. You've got to make very sure that it's well sealed, so we'll make sure the top is screwed on tightly. We've got a good seal. Now this will have to incubate for 24 hours at 68 degrees or 20 degrees Celsius. And the way that we are doing that to control the temperature is in our growth chamber that we normally use for germination testing on seed. But it's nice because it does have controlled temp and a timer on it. And so we will go ahead and stick this in the growth chamber and wait for 24 hours. We have some samples that have been incubating for 24 hours, and we're going to go through the process of reading and interpreting that data. So we have one jar for each of the soil samples that we described earlier, from the tilled hay field to the new hay seeding, new bale grazing, and established bale grazing. When you look at the jars, right off the bat you can see some difference in the color. First off, they've obviously changed from that nice royal blue that we started with 24 hours ago, and they're different gradations of sort of this blue to yellow spectrum. The sample closest to me here, the tilled hay field, is sort of bluish yellow or kind of a dark mustardy color. As we move our way over towards the more intensive bale grazing practice that we would expect to increase organic matter, microbial activity, and things that we're looking for in this test, you can see a bit of a spectrum going to more yellow in color. So now what we have to do is actually read these paddles and see what numbers we come up with. So at 24 hours, we'll open the jars, pull out the paddle, and it's going to go face up into the reader. And then we hit this low CO2 for the first method. And we hit the read button. Move on to the next one. So this is our new hay seeding. And read. We got a color of 4.60 and CO2 of 115. Our new bale grazing came up with 4.89 on the color and 148. Current bale grazing. I would expect this to be the most biologically active or healthiest sample. Of 5.22 on the color and 198 parts per million. To review, on the color scale across our spectrum of samples going from a tilled hay field to pretty intensive bale grazing, we went from 4.26 to 4.6 to 4.89 to 5.22 on the color. And on the CO2, from 85.5 to 115 to 148 to 198 parts per million. Now what does that mean as far as the health of these soils? Our lowest sample was 4.26. So even on the low end, at 4.26, we are looking at soils that are in the medium high range of soil biological health. As we move up to our established bale grazing at 5.22, we're nearly at the top of the scale. And 198, we're actually in what they're calling the excessive biological health range. When we think about organic matter in soil, there's a lot of benefits, right? We've got water holding capacity, we've got nutrient holding capacity, we've got carbon sequestration in the soils. We've got the ability of those microbes to break down those carbon molecules and release nutrients like nitrogen. All those things are beneficial. At the same time as that process occurs, we are releasing CO2 from the soil into the atmosphere, which can contribute to climate change. So it's a delicate balance of sequestering carbon in soils, using them as that carbon sink, keeping that system cycling and healthy, while trying to minimize the emissions that are coming out of our farm systems and contributing to climate change. So it's great to see that the farm that we're working with out there in Dedecker is doing a really nice job managing their soils. And we've got a farm system that is designed to take care of soil health in that we are doing things like pasture, like perennial cropping and hay production, and managing our manure and our feeding in the bale grazing practice to really try to address some of our soil health concerns and increase soil carbon. So it's apparent that that farmer is really doing a great job managing their soil. To say it's excessive, maybe in that particular spot in the field, we're actually maybe overdoing it, right? Or maybe got a little bit too much manure in one place, a little bit too much residue in one place, and maybe that bale should have been a little smaller, or the next bale should have been moved a little bit further away. But overall, we're seeing great things in terms of the soil health in these samples.
That's what's up at Upright. Hope to see you again next time.
I've been everywhere.